Welcome to this lecture on oogenesis, the fascinating biological process responsible for making a female gamete. Imagine the incredible complexity required to create the very foundation of human life. In this session, we are going to explore how the female body carefully prepares and nurtures a single masterpiece, the egg cell. We will build our understanding from the ground up, looking at the biology, the mathematics of genetic reduction, and the real-world medical applications. Let us dive right in. To begin our journey, let us think about the ovary as a highly exclusive, specialized factory. Now, why does this matter? It matters because unlike male sperm, which are mass-produced by the millions every single day, the female body takes a radically different approach. It focuses all of its resources on creating just a few highly viable eggs. This process is incredibly resource-intensive. It actually begins before a female is even born, pauses for years during childhood, and only resumes during puberty. Understanding this biological investment helps us appreciate the sheer value of a single human egg. Now that we understand the ovary's role as a specialized factory, let us look at the formal definition of what is actually happening inside. Oogenesis is the biological process where female germ cells, known as oogonia, differentiate and mature into fully functional ova, or eggs. This is not a simple division. It involves both mitosis, which multiplies the cells, and meiosis, which halves the genetic material. What makes this truly unique is the unequal cell division. One cell receives almost all the cytoplasm and nutrients to sustain a potential embryo, while the other becomes a tiny, non-functional polar body that eventually degrades. Building on that definition, let us break down the timeline of this incredible process. Oogenesis does not happen all at once. It is divided into 3 distinct phases. First is the multiplication phase, which occurs entirely during fetal development, before the female is even born. Next is the growth phase, where the primary oocyte accumulates the massive amount of nutrients it will need. Finally, we have the maturation phase, which spans from the onset of puberty all the way to menopause. This highly regulated cycle ensures that eggs are released only when they are perfectly ready. Understanding these phases naturally leads us to ask, why is this so critical in the real world? The importance of oogenesis cannot be overstated because it is the fundamental prerequisite for human reproduction. The egg provides not just half the genetic blueprint, but all the initial cellular machinery and mitochondria for the embryo. In reproductive medicine, doctors evaluate a woman's oogenesis cycle during in vitro fertilization or when assessing fertility. Furthermore, errors during these meiotic divisions are the primary cause of chromosomal abnormalities, like Down syndrome, making this knowledge absolutely vital for genetic counseling. Since we just mentioned meiotic divisions, let us look at the mathematical representation of this genetic reduction. Conceptually, meiosis must reduce the genetic material by exactly half, so that when a sperm and egg fuse, the resulting baby has the correct amount of DNA. The formula for this is n sub O equals n sub S divided by 2. Let us define these symbols. N sub O represents the number of chromosomes present in a fully mature human egg cell. N sub S represents the total number of chromosomes found in a standard non-reproductive human body cell, like a skin cell. The 2 in the denominator represents the 2 rounds of meiotic division. If the starting variable n sub S were to increase or decrease due to a mutation, the resulting n sub O would change proportionally. Now that we have our genetic reduction formula, let us see this with a concrete example. Imagine a reproductive endocrinologist is studying a healthy human female's ovarian tissue. We are given that a normal human somatic cell contains 46 chromosomes, and we are asked to find the number of chromosomes in the final mature ovum. We choose our reduction formula because meiosis cuts the genetic payload in half. We substitute 46 for n sub s and divide by 2. The math gives us a final answer of 23 chromosomes. Physically, this means the egg is perfectly prepared to meet a sperm cell, which also contains 23 chromosomes, to create a healthy embryo. So we've seen how the chromosomes are halved, which brings us to another unique biological rule of oogenesis: unequal cytokinesis. Why does this matter? Because it explains the physical size difference between the cells produced. Unlike spermatogenesis, which yields 4 functional sperm from one primary cell, oogenesis sacrifices quantity for quality. One primary oocyte yields only 1 functional ovum, hoarding the vast majority of the cellular cytoplasm. The remaining genetic material is cast off into up to 3 non-functional polar bodies. This monopoly on nutrients is essential to support the early stages of an embryo's life. This biological rule of unequal cytokinesis leads us directly to the mathematical formula for calculating cellular yield. Conceptually, since one primary oocyte only ever produces one viable egg, the relationship is a simple 1-to-1 ratio. The formula is V equals n sub p times 1. Here, V stands for the total viable ova produced. N sub p represents the starting number of primary oocytes. Because the multiplier is exactly 1, any increase in the starting number of primary oocytes results in the exact same increase in viable eggs. This mathematical relationship perfectly reflects the biological focus on quality, showing why female fertility is so heavily dependent on the health of each individual starting cell. Before we move on, let us apply this yield formula in a clinical setting. During an in vitro fertilization cycle, a fertility clinic stimulates a patient's ovaries to bring 8 primary oocytes to full maturation. We are asked to calculate the total number of mature ova produced. We use our viable ova formula because of the 1-to-1 biological ratio we just discussed. We substitute 8 for our starting number, n sub p, and multiply by 1. The final answer is exactly 8 mature ova. Physically, this tells the embryologist exactly how many functional eggs they have successfully retrieved for potential fertilization. Now that we have calculated the viable eggs, we must also account for the waste cells, using the formula for polar bodies. Conceptually, the egg needs to shed half of its DNA without losing its valuable cytoplasm, and it does this by creating genetic trash cans called polar bodies. The formula is P equals n sub p times 3. In this equation, P represents the total number of non-functional polar bodies generated. N sub p is again the starting number of primary oocytes. The multiplier is 3 because each complete meiotic cycle produces up to 3 of these tiny cells. If the number of starting oocytes increases, the number of discarded polar bodies increases at 3 times that rate. Let us continue with our previous clinical example to calculate these discarded cells. We are still working with the same 8 primary oocytes from the IVF cycle, but now we are asked to find the total number of polar bodies generated. We choose this specific formula because each primary oocyte yields up to 3 polar bodies during meiosis. We substitute 8 for n sub p and multiply by 3. The final answer is 24 polar bodies. Physically, this calculation shows that a significant amount of cellular material is sacrificed and naturally degraded by the body, all just to create those 8 perfect eggs. To tie all these concepts and calculations together, let us walk through this flowchart of the oogenesis stages. At the very top, we start with the diploid oogonium, containing 46 chromosomes. Following the arrow down, it becomes a primary oocyte. During meiosis I, notice how the cell divides unequally into a large secondary oocyte and a tiny first polar body. Moving down to meiosis II, the secondary oocyte divides again, producing the large, mature haploid ovum with 23 chromosomes and a second polar body. Meanwhile, the first polar body also divides. This diagram beautifully illustrates the mathematical reduction of chromosomes and the physical reality of unequal cell division. We have covered the full topic of oogenesis, so let us review our key takeaways. First, oogenesis is a highly specialized, resource-intensive process designed to create a female gamete. Second, it relies on meiotic division to perfectly have the genetic material, resulting in an egg with 23 chromosomes. Finally, through unequal cell division, the body prioritizes quality over quantity, producing one massive, nutrient-dense egg and several disposable polar bodies. You now have a solid understanding of this biological marvel and how human life begins. Great job!